Microbiology and rightly Professor and Head, Department of Laboratory Medicine. The topic which I am going to teach you today or talk about is biomedical waste management. I think a topic which is entirely new to you. Just because you are in a medical college, you are supposed to know about this, what exactly it is. So in this lecture, we will know about definition and characterization of biomedical waste, what are the health impacts of BMW, legislative, regulatory and policy aspects of biomedical waste, handling, storage and transportation, and then how can we treat and dispose it. Okay. So this is a definition for biomedical waste. Biomedical waste is any waste which is generated during the diagnosis, treatment or immunization of human beings or animals, or in research activities pertaining to or in the production or testing of biologicals and categories. So if you see at the definition, if you look at the definition, there are many things which are present. So if we add the during the diagnosis, all the laboratories come under it. Treatment, all the hospitals or nursing homes come under the definition. And immunization, even if you are running an OPD where you are immunizing the young children, that will also be the part of it. Then there are research activities, particularly regarding the biologicals. So everything which is being generated over there is biomedical waste. So as I said, biomedical waste is generated in hospitals, nursing homes, clinics, medical laboratories, blood banks, animal houses, etc. Such a waste can also be generated at home if healthcare is being provided there to a patient, like injection or dressing material. So healthcare waste is defined as the total waste that streams from a healthcare facility. There are two basic categories out of it. Healthcare general waste that constitutes around 75 to 90%, and there is healthcare risk waste that is around 10 to 25%. So out of this 100, this is only 10 to 25% which is risky to us as well as to the environment. Now, so what will happen if we mix both of them? Means if we do not care about what kind of waste is being generated and we put into a single kind of dustbin. So everything becomes risky. So healthcare general waste includes paper packaging, plastic packaging, food preparation and other items that have not been contaminated yet. And these are the different categories of healthcare risk waste, that is infectious waste, pathological waste, sharps, pharmaceutical waste, genotoxic waste, chemical waste, waste with having a high content of heavy metals, pressurized containers and radioactive waste. These all things are risky in nature as well as infectious also. So infectious waste is suspected to contain pathogens like bacteria, viruses, parasites or fungi in sufficient concentration or quantity to cause disease in susceptible host. Examples are cultures and stocks of infectious agents, waste from surgery and autopsies, waste from infected patients in isolation wards, hemodialysis and infected animals from laboratories. Pathological waste consists of tissues, organ, body parts, human feces and animal carcasses, blood and body fluids. Within this category, recognizable human or animal body parts are also called anatomical waste. Sharps, these are the items that could cause cuts or puncture wounds, including needles, hypodermic needles, scalpel or other blades, knives, infusion sets, saws, broken glass and nails. Whether or not they are infected, such items are usually considered as highly hazardous healthcare waste. Now then comes pharmaceutical waste that includes expired, unused, spilt and contaminated pharmaceutical products, drugs, vaccines and sera that are no longer required and need to be disposed of appropriately. Genotoxic waste, this is highly hazardous and may have mutagenic, teratogenic or carcinogenic properties. Genotoxic waste may include certain cytostatic drugs, vomit, urine or feces from patient treated with cytostatic drugs, chemicals and radioactive material. So oncology department. In a specialized oncological hospital, genotoxic waste, which is containing cytostatic or radioactive substances, may constitute as much as 1% of the total healthcare waste. That is a too high number to cause significant harm to the people who are serving over there. For example, if a powdered ampule or a glass vial is broken in an oncology unit, you are supposed to immediately evacuate the area. Why? Because if you inhale it, or person who is serving over there, if they inhale it, it may cause cancer to them also. Then comes chemical waste. They consist of discarded solids, liquid and gaseous chemicals, for example, diagnostic and experimental work, and from cleaning, housekeeping and disinfecting procedures. Chemical waste from healthcare may be hazardous or non-hazardous. It is considered to be hazardous if it has at least one of the following properties. So they are toxic in nature, they are corrosive, like acid of less than 2 pH and bases of more than 12 pH. They are flammable, they are reactive, like explosive, water reactive or shock sensitive, and they are genotoxic. So the types of hazardous chemicals used most commonly in maintenance of healthcare centers and hospitals are formaldehyde, photographic chemicals, solvents, organic chemicals and inorganic chemicals. Formaldehyde कहाँ यूज होता है? अभी कहाँ सामना हुआ formaldehyde? कहाँ क्लासेस शुरू हो गई ना? कितने बजे से शुरू होता है सुबह डिसेक्शन? अभी शुरू नहीं हुआ? ओके। जब शुरू होगा तो एक चीज नोटिस करना. So those who reach first in dissection hall, for example, if it starts at 8:30 and you are reaching there 8:25, quite eager to do the dissection. When they open the gate, what to say, formalin vapor will be filling the dissection hall completely. Because throughout the night they put the cadaver into the formalin drums, and when they take it out, a lot of formalin vapor is there. It will sting your eyes, your nose. So be careful, because rather than the fear of cadaver, I think it is the formalin which causes the more trouble. Cadaver सबको अच्छा नहीं लगता, मेरे को भी नहीं लगता था, but to just keep that aversion aside, close your eyes, put both of your hands in the cadaver for 1 minute, खत्म. उसके बाद फिर दिक्कत नहीं होता. But formalin साल भर परेशान करेगा. So for formalin, you can do just one thing. Whenever you come out of the dissection hall, when you are there, I can't tell you anything because sometimes they allow the mask or sometimes they not. But whenever you come out, just wash your eyes and nose. That's all. Face wash kar lo. Formalin is over. How can you? Now suppose if there is a spill of formalin, what will you do? कभी कुछ फैल गया. इतने बड़े हॉल के लिए if we spill just 1 liter or 1.5 liter of formalin, that is sufficient. We'll all have to run out. So how can you neutralize the formalin vapors? Have you heard of ammonia? So ammonia itself is having fumes which are quite irritant in nature. But ammonia neutralizes formaldehyde gas very effectively. So if you have spilled 1 or 1.5 liters of formalin over here, just take 1 liter of ammonia in 4 bowls, keep it at 4 different points, switch on the fan. The vapor of ammonia will neutralize the vapor of formaldehyde. But it does not mean that you are going to take ammonia from biochemistry lab and going to... Don't do that. Okay, that is only for the case of spill. Now waste with the high content of heavy metals like mercury waste, cadmium waste and lead. So मर्करी कहाँ यूज होता है हॉस्पिटल में? सबके घर पे है थर्मामीटर? कितने दिन हुए एक मर्करी वाला थर्मामीटर यूज करते हुए? डिजिटल यूज करते हैं नहीं करते आजकल? Anything else other than thermometer which used to have mercury in it, mercury column? BP apparatus. Scientific name for BP apparatus क्या बोलते हैं उसको? क्या? चलो, सबको आता है. So nowadays they do not use mercury in BP apparatus also. They are also digital. Lead commonly used in batteries. Meaning, see, in a household, the amount is not that high, but in case of a bigger institution where a lot of batteries are being used, that can be a very significant number of lead which is being thrown away. And lead is a very heavy metal which pollutes the land as well as the water also. So to summarize this, biomedical waste, non-hazardous or hazardous. Hazardous could be infectious or other. Infectious may sharps, non-sharps, plastic disposables and liquid waste. In hazardous, radioactive, discarded glass, pressurized containers, chemical waste, cytotoxic waste, incineration, ash. So biomedical waste falls in a very different category in comparison to the domestic waste. So there was a rule, Biomedical Waste Management and Handling Rules 1998, and there they gave the 10 numbers 1, 2, 3, 4, 5, 6. They categorized by the number. They specified what will fall into that numbered category. So for example, category number 1 used to be human anatomical waste. So if you do amputate a limb in surgery, if you are throwing a limb from your anatomical dissection hall, that will go into the category 1. And according to the category, there was a treatment, either incineration or deep burial, for example in this. So category 2 used to be the animal waste, category 3 microbiology and biotechnology waste, category 4 waste sharps, category 5 discarded medicine and cytotoxic drugs, category 6 soiled waste, category 7 solid waste, category 8 liquid waste, category 9 incineration ash, and category 10 is chemical waste. And they suggested that these categories should be discarded into a color coded bin. So there used to be red, yellow, blue and black, right? The problem with this system was some categories were common to the different color bags. So category 1 and 2 could be thrown either in a red or yellow bag. So by seeing the bag itself, you will not be able to differentiate what should I do with this. So to overcome these problems, they came with the new rules which are prevalent now, that is Biomedical Waste Management Rules 2016. And instead of giving a number to a category, they have given it a color. So there are 4 colors now: yellow, red, blue and white. Now in yellow, they are saying they can throw human anatomical waste, animal anatomical waste, soiled waste, discarded or expired medicine, microbiology, biotechnology and other clinical laboratory waste, chemical waste and chemical liquid waste. So they will use a yellow colored non-chlorinated plastic bags. Red category contains only one thing. It's a contaminated waste which is recyclable, right? So a contaminated dressing will go to yellow. A contaminated tubing, plastic tubing, that will go to red, because plastic tubing can be recycled. So for that we use red colored non-chlorinated plastic bags which are having thickness equal or more than 50 microns. Then white category, it contains waste sharp including metals. So for that they take white colored, translucent, puncture proof, leak proof, tamper proof container. So this container is having a lid, there is a hole in the lid. Once you fill it 3/4, you seal the lid and then it goes for the deep burial. Then there is a blue category which contains glassware or metallic body implants. So for that we can use a puncture proof, leak proof boxes or containers with blue colored marking. So we can make a box like that, or we are having in market available blue boxes like this. So as I said, these are 4 categories. Now point is, what will you like to do if a healthcare facility is generating waste? So first of all, you will mix all the waste and later on you are going to segregate it, or you are going to segregate it at the point of production itself. So the best thing will be, because once you mix it, you can segregate it, but now the whole means entire waste is contaminated. Second thing, it put risk to the person who is segregating it. So it's better we put a 4 color coded bins in every ward, and then accordingly we can throw waste into it. So when you go to your internship, just keep in mind what happens with the time. Now you people अभी gloves discard करोगे तो ऐसे उतार के डालोगे, जब internship में जाओगे तो ऐसे डालोगे. So that happens. It's something which is natural. But the point is, at least aim in the right box. कि मैं मना करूंगा aim नहीं लगाओ तो वो तो होगा नहीं. So at least go for the right box. Because if you throw dressing in a black one, that black bag is going to be treated as a domestic waste. No one is expecting there will be a sharp or a blood sample into it. So they are unaware about it, and you are putting that person at risk by your act. Right? So segregate. Biomedical waste should be segregated, preferably at the point of generation. Color-coded bags and bins are used for that, and it helps in safe handling and transportation. So as I said, for the 1998 rules, in yellow we could have put category 1, 2, 3, 6, and in red category 3, 6 and 7. As you can see, category 3 and 6 is overlapping in both the bags. Category 7 is overlapping between red and blue, and that was the problem. Now once you have segregated waste, what happens exactly? So at a hospital basis, we collect the waste in the ward, and in the morning there is a team of persons who go to every ward, take that bag, put it into a closed lid container, means a trolley over there, and they transport it to the biomedical waste storage facility. As a hospital, we do not treat our waste by our own, but we have given it to a third party. So that name is Instromedics. Probably through the entire Rajasthan, that is the only one which is available. And they take it from us, they treat it at their own site. But the point is, if our waste is not treated or handled properly, यहाँ से लेके उनके treatment तक, वो responsibility is of institution. So that is the liability under the law. Now suppose, to explain it, there is a waste, there is a sharp container which is taken from Mahatma Gandhi by Instromedics. They were going by their van and it fell down on the road, and all the sharps came onto the road. There were some people who were walking barefoot and they all got injury due to it. So who is liable in this case? Instromedics or Mahatma Gandhi? This is Mahatma Gandhi. Mahatma Gandhi is responsible from the production of waste till its proper disposal. Right? So these are the different methods available: double chambered incineration, autoclaving, microwaving, hydroclaving, and chemical treatment. So double chambered incineration is, there are two chambers. In first, they just heat it up to a certain temperature. So primary chamber, there is a less temperature, 800 degrees Celsius only, and it gasification or pyrolysis happens in this primary chamber. So the gas which originates from the primary chamber goes to the secondary, which is having a higher temperature, and complete burning takes place over there. Disadvantage: toxic gas production. Autoclaving, you can do it, but you will require very large volume autoclaves for that. You will also need biological indicator testing for every 40 hours of use, logbook require, and regular maintenance. Cycle will be 121 degrees Celsius, 15 to 20 lobes pressure for 60 to 90 minutes. Usually that cycle is 121 degrees Celsius, 15 lobes pressure and 15 minutes. Transport waste to the autoclave in closed bags and leak-proof container. Microwaving takes place at 97 to 100 degrees Celsius. Cycle is of 45 minutes. It's not to be used for the cytotoxic, hazardous or radioactive waste or contaminated animals, carcasses, body parts and large metal items. And probably this could have been the answer to all these troubles with other methods, but it never gained the popularity. I don't know why. So this is hydroclave process. Hydroclave के बहुत सारे फायदे होते हैं. The waste which comes out of it is having a very less volume in comparison to the original one. There are no gases emitted in the process, and it basically can work like a fertilizer also. So hydroclave can process bagged waste in ordinary bags, sharp containers, liquid containers, cardboard containers, or metal objects. So first of all, you put it into the hydroclave. There are powerful rotator which mix the waste and breaks it into small pieces, and there is a steam which fills the double wall, as you can see that blue one, of the vessel and heats the vessel interior. The liquid in the waste turns to the steam which further sterilizes it. After 20 minutes, the waste and the liquids are sterile. Now the vent is opened and the vessel is depressurized. Steam heat and the mixing continues until all the liquids are evaporated and the waste is dry. The unloading door is opened. The mixer now rotates in the opposite direction, so angled blades on the mixer can push it down, and the dry sterile waste can be fine shredded further or dropped in a waste disposal bin. So that's the benefit. It was initially fully filled, and now that is the amount which is coming out. So dry waste, regardless of its original water content, low odor due to the dryness, volume reduction up to 85%, weight reduction up to 70%, accepted as harmless waste. And then last is chemical treatment. It's acceptable for liquid material if done correctly. Add full strength household bleach to final concentration of 5000 ppm available chlorine. Mix. Contact time should be at least 30 minutes and pour down drain to the sanitary sewer. So that was about biomedical waste. If you are having any question, you can ask.